Okay, so welcome everyone to our April edition of the Chemaraderie. We've had a month's break off of this due to the fantastic Baylis presentation we had last month by LD. So yeah, we're back today and for next month as well. Looks like we're getting reverb. So I'd like to start by acknowledging the traditional custodians of the land that we're meeting on today and pay my respects to their elders past, present and emerging. And so yeah, on to the main event for today. So today we've got Dr. Adam Martin presenting. For those who were here in October, he was one of the panelists for the Wire Industry event that we had. Obviously, he's also here. So we thought we'd let him come and give a talk and tell him all about his actual job and not just why he went into industry. So he's the Director of Chemistry at PYC Therapeutics, and he's had 10 years experience in peptide therapeutic, encompassing therapeutic design, manufacturing, display technologies and conjugates. He currently leads PYC Therapeutics manufacturing and chemistry operations, in addition to its peptide discovery platform. He's published over 50 papers and several patents, and been awarded $5 million in research grant funding and numerous prizes. So I would list them all, but it looks like there's quite a few, so well done.
No problem. So today I'll talk to you a little bit about just my journey in the academic space in biotech. I look to have a bit of a chat to you guys about the peptide space, peptide therapeutics, and what we do at PYC Therapeutics. This is an interactive presentation, right? So if you have questions, don't hold them until the end. Feel free to interrupt me at any time, heckle me, questions, whatever, right? That's fine. It's an interactive session. And a disclaimer, I'll be talking a little bit about experiences in industry. That's my experience in industry, my experience in biotech. Your experience may differ from what you see on the screen. So quick show of hands in terms of the split between academia and industry here. How many folks are in the academic space at the moment? Okay, industry space? And sent here by PYC to spy on me? Good, Mark in the back. All right, fantastic. Great, so Mark will censure me if I say anything that has not previously been released in the public domain. So look out for that if I slip up there. Great. So a little bit of an outline of the talk that I'm going to give today, and Terry, please feel free to throw something at me if I'm running over time, so absolutely. A little bit on my background and journey, just a crash course in peptides, right? We'll talk a little bit about peptides today, so I'll give you guys a crash course in peptides, a little bit about self-assembling peptides, which is a lot of the work that I did across my time at UNSW, at Macquarie as well, and then getting into biology, right? So I'm a chemist by training, and I was not a huge fan of biology when I started, but it turned out okay in the end. Venturing to the dark side of industry, and then I'll tell you guys a little bit about PYC and I guess in my experience, the step change between what makes a good paper versus what makes a good regulatory submission, right? And then a little bit of myth busting at the end. Okay, so I actually started 20 years ago here, which is a little bit depressing now that I think about it. I'm still getting feedback from that. Did nanotech at Curtin Uni, and I sort of went into a PhD in chemistry because, I'll be honest with you guys, I wasn't smart enough to do physics. I wasn't smart enough to do the maths, and I had a first year chemistry lecturer by the name of Marie Baddock, who did a demonstration, I think, at the front of a lecture theatre doing it was a copper ammonia demonstration, right? So she had copper sulfate in a conical flask, added some ammonia, formed this milky blue solution, and then added more ammonia, and it went to this beautiful dark royal blue, right? And I'm a simple person. Give me a colour change. I'm very happy. And so that was it. I was hooked on chemistry, and that's sort of the way that I went. The lesson that I learned during my degree and during my honours year was time management, right? I was doing, back in those days at Curtin, it was 50% project, 50% coursework. I was also working 40 hours a week and doing extracurriculars as well. So it was a good crash course in time management. And then I did my PhD at UWA. So I went to UWA, moved over, did a PhD, oh that's clever, in supramolecular chemistry with Colin Raston. Colin For those of you who don't know Colin, this is the same scientist that was awarded an Ig Nobel Prize on how to unboil an egg, right? So he had a lot of ideas, did Colin, and the thing that he taught me more than anything else was how to write. How to write a paper, right? How to sell your research, how to communicate what you're doing. That's something, and all of the lessons that I've learned throughout my career sort of have come in handy in my current role. So Colin taught me, amongst other things, how to write a paper, but I didn't really know a lot about chemistry. I'll get to that in a sec. After I finished my PhD at UWA, I spent a bit of time as Perth's most overqualified sandwich artist. So I did a bit of an RA, I sent CV after CV after CV, applied for jobs. I probably would have cold emailed 50 to 60 folks looking for a postdoc. The 40 hours a week that I was doing up here, up there, was actually at a Subway shop. So I knew the guy, and at the end of the day, sometimes it's just nice to have to relax and not worry about writing papers and reading papers, and so that was a nice little jaunt. But eventually I got a postdoc over at the University of Nottingham, working on metal organic frameworks with a professor called Martin Schroeder. That was an experience, right? Because Colin was a very hands-on supervisor, right? You'd go into his office, you'd meet him whenever you could, he'd give you input, and there was a lot of back and forth. Martin is a group of 60 people, right? He was never ever in, always traveling at conferences, you would never ever see him. I think I was there for 2 years and I had 2 meetings with him. So it was very much, the group was run by the postdocs, a different dynamic, and it was very sink or swim. And that is where I learned chemistry. Because when I was doing my PhD, all of the reactions that I did had been optimized for me by the guy that came before me, right? So I just had to roll in, do the chemistry, get some crystal structures, do a little bit of NMR work back in the day where you had to climb underneath the NMR and manually adjust the tuning and matching, and got a few papers. But here, this is where I actually learned, I would call it proper synthetic chemistry, right? To the point where I asked a postdoc at the time, I said to him, can you help me run this column? And he said, yeah, why is it a problematic column? I said, no, I've just never run a column before. And he looked at me and he goes, did you get your PhD out of a cereal box? Which, I mean, fair comment, to be honest. But once we got over that, him and I became very good friends, and we remain friends to this day. Nottingham was great, right? I loved it. I loved being in the UK, but the job was going nowhere, right? Unless in that group, unless you're one of the golden children, right, it was hard to get papers. And papers, as you guys know, that's the currency of an academic. You need to build up your resume, your CV by putting out papers. And there was a 3-year backlog on papers in this group. So it was never ever going to happen for me there. So I'd reached out to a few folks back in Australia, and eventually I landed a postdoc with our fearless RACI president, Parley Thordarson, Professor Parley Thordarson. And Parley had actually reviewed my PhD thesis, so I knew him from that particular role. And I possibly gave one of the world's worst interviews for that role. Right? And I remember this, and he said to me, I won't do my Parley impersonation because this is being recorded, but ask me about it later. So he said to me, give me 3 slides for your interview. I want to know, what have you done in the past? What are you doing now? And if you get the job, what are you going to do in this group? So I told him the first 2, and for the third presentation, for the 3rd slide, his group had just published a paper on organogels in JACS. And so I said, hey, look, you've got this paper in JACS, and you could do this structure activity, and you can look at anion-gelators, and you can build on this work. And I get through this 5 minutes of telling him what I'm going to do, and he said, yeah, we don't work on those molecules anymore. We've moved on to peptide hydrogels, actually. And so I thought, well, look, it was nice. It was nice of him to give me an interview. But there's no way I'm getting this job. And he calls me a couple of days later and he says, congratulations, you got the job. And normally when someone says that to you, you say, fantastic, thank you. I'm really excited. And my response was, really? How bad were your other applicants, Parley? But no, it was good. And actually a lot of that is he knew that I could write from my PhD thesis, and in his group he just needed someone who could do research and write papers and help him out on that front. So that was good. I learned a lot about biology there, and I hated it. I wanted to leave. I got there, I started looking at peptides, and I considered myself a chemist, a synthetic chemist, and I was like, what is this peptide stuff? What is biology? The first meeting that I ever sat in on in Parley's lab, at the end of the meeting, I raised my hand and I said, hey, I just got a real quick question. I might have missed it, but what's GFP? And everyone looked at me like, oh God, where did we find this guy? Is this the guy you hired? This was in a protein group meeting, right? All they talked about was GFP this and folding that. So in time, I stuck it out. I got to learn a little bit about biology, and that stood me in good stead for the next role, which was the NHMRC fellowship that I got in 2015, right? This is a right place, right time story. And everyone who's in academia knows that you need a little bit of luck sometimes, right? You can have an amazing grant proposal, you can have an amazing paper. Sometimes you just need someone to get out of the right side of bed in the morning, right? And this was sort of right place, right time. This particular fellowship, it was a fellowship in dementia. And the call was for people who weren't in dementia, who were 4 years or less post-PhD. So I ticked both those boxes, but it ruled a lot of people out for that fellowship. And it flew under the radar a little bit. So I was lucky enough to get one of those, and full credit has to go to Parley for pulling me into his office and saying, hey, I think you should apply for this fellowship. And I remember saying to him, it's dementia. We're chemists, right? But he put me in touch with a guy at UNSW, Lars Zidner, who was working in the space, and eventually I became a group leader in Lars's lab. So Lars has a very well-constructed, well-funded lab that was at UNSW. He moved to Macquarie. I moved to Macquarie with him. I took that opportunity to be a group lead and basically set up a lab there. So set up a peptide synthesis lab, and it was kind of there where I learned a little bit more about the logistics of running a lab, right? The people management side of things, the sort of all of the space requirements. I've sat in on more meetings about HVAC than I would ever want to repeat again. So all of this has kind of fed into what I do at the moment at PYC. So I learned a lot of lessons along the way. And at PYC, I think the biggest lesson that I learned, and I'll touch on it a little bit later in this presentation, is the value of time, right? You never, in academia, and maybe it's just me, I never thought about time as a valuable commodity. Everything was always money, right? And you're always trying to scrimp and save. But here at PYC, time is probably the most valuable thing that we have here. All right, peptides 101. What is a peptide? So really, peptides are fairly simple molecules. N-terminus, a sequence, and a C-terminus, right? Pretty straightforward, but the peptides are interesting because the function of a peptide is very dependent on its structure, right? The way that I like to think about a peptide is it's like a rope, right? You can have a sequence that, you can have a rope, and the rope can be only useful for mooring a boat to a jetty when you tie it in the right kind of knot. A peptide is exactly it's exactly the same thing. The peptide function as a protein or whatever only happens when it's folded the correct way. All right, so the function is related a lot to its structure, and the structure is dependent, obviously, on its sequence, right? But structure-activity relationships, or SARs, in peptides are not always that straightforward, right? You can change one amino acid in a peptide or in a protein, and you can completely obliterate the function of that particular protein or peptide. I used to work a lot in self-assembling peptides. You would change one amino acid from a phenylalanine to an alanine, and something that gave you these beautiful, robust gels that were amazing for growing cells on, all of a sudden were watery messes that just were unworkable, or insoluble messes that you just couldn't ever purify without clogging a HPLC and really making the rest of your research group quite angry at you. So in terms of structure-activity relationships, peptides can be a little bit like the Wild Wild West, right? They're a bit hard to predict, and this is why tools like AlphaFold and Rosetta Fold and MSA Fold are so useful, right? That's why they've been such a game changer in this particular space. So I worked a bit on self-assembling peptides when I started at UNSW. And as I said to you guys before, I hated it. I was coming from the world of metal organic frameworks, right? Schlenk line synthesis, putting it into a solver thermal reaction where you put your ligand, your metal salt, DMF, whatever it is, in a sealed vial, you heat the crap out of it overnight, and you come in the morning and hopefully you've got some beautiful crystals on the wall of your vial, right? You then get the crystal structure, you do some powder X-ray diffraction, you do some gas absorption work, right? You get these beautiful, beautiful PXRD patterns, lovely sharp peaks, lovely crystal structures, right? And all of a sudden I was working with hydrogels which were an amorphous mess, right? So you'd gone from X-ray diffraction to neutron diffraction, right? Now, as a chemist, it's pretty easy to make sense of this, right? I mean, X-ray diffraction, these are lattice spacings within your crystal. You can reconstruct that and build up your crystal structure. What the heck is that? That's a water slide, right? That's not you can't get any useful information out of this. So, and the first thing that Parley asked me to do was write applications for neutron scattering beam time. I told him I just didn't agree with the technique. That wasn't really an option, so I sat down and I wrote those beam time scattering applications. But you can actually get a lot of information from these sorts of neutron scattering curves once you know how they work, right? A gel really is just an intermesh tangle of fibers everywhere, right? Think of it like a ball of yarn that's just binding water together, right? And what these actually tell you have a little hump here, and that hump tells you a bit about the diameter of the fibers of the yarn, and this tells you about the pore size in the network there. So that is, once you sort of know a little bit more about it, these curves become a little bit less boring and a little bit more informative. But I guess the lesson is, I wanted to quit after the first week. After the first month, I still wanted to quit. The only thing that stopped me from quitting was because what other job was I going to get? I didn't know anyone that owned a Subway in Sydney. I only knew them that owned Subways in Perth. So I stuck it out. And I thought, maybe I'll grow to sort of build my bio expertise. And that's kind of what happened. So I'm glad that I stuck it out. Okay, now, in Parley's words, how the heck do you define a hydrogel? He would say. Well, if I showed you what a gel looks like, if I showed you 4 vials, right, and I inverted them all, you could easily tell me this is a gel, this is not a gel, right? Things like your toothpaste that squeezes out of a toothpaste tube, or if something holds its shape when you invert a vial, you would tell me that's a gel, right? Something that's like a cheesecake, you flip it upside down and it doesn't gloop out everywhere, that's a gel. It's actually hard to define a gel though sometimes, other than, yeah, I see it, it looks like a gel. The way that we used to define it was, it's basically crystallization in one dimension. That's all that gelation is. It's frustrated crystallization. It is a monomer or something like this which has order in one dimension. It's stacking in a certain way, but it doesn't have enough order to stack in a regular ordered manner in 3 dimensions. Right? So instead, it forms these fibers, and the fibers can bundle up, or the fibers don't bundle up, and then they entangle with each other. And when they entangle with each other, they start to entrap a solvent, right? And it can be an organic solvent. So I know Emily did a lot of work on organogels in her PhD. I did a lot of work on hydrogels in my postdoc. Your solvent of choice, right? And like crystallization, it's a balance between solubility and insolubility, right? If you want to crystallize salt, or if you want to crystallize sugar, or whatever it is, you heat something to a point of supersaturation, right? And it crystallizes. Or you change the solvent. You put it in a good solvent, you introduce a bad solvent, right? That's crystallization. Gelation is exactly the same, right? We used to take these molecules, we used to dissolve them up in base, and we used to add a gelation trigger, which was a slowly hydrolyzable acid. It would slowly drop the pH, and you would slowly protonate these things. You would slowly protonate the fibers, which would begin to self-assemble, reduce the repulsion between the fibers, and they would form a gel, right? Same. It's just, and it was just required a bit of change in the way that I was thinking about what a gel is. So some work that I did during my postdoc, I look back at it now and it's very narrow, right? It's narrow work. It's sort of like it's the classic, I'm going to take a peptide here that has the same sequence, or a diphenylalanine sequence here. I'm going to change the capping group on the N-terminus. I'm going to use a somewhat hydrophilic indole group. I'm going to use a somewhat hydrophobic carbazole group. And I'm going to show you that the mechanism of self-assembly is different. Right. For the hydrophilic group, what actually happens is you have these solvated fibers which form bundled gels, and you get these really thick fibers that you see by TEM that's really strong hydrogel. Right? With this hydrophobic amino acid here, you actually get these preformed fibers. The nice thing here is that you form these so this is about 50 to 100 nanometers. This here is sort of in the order of 3 to 4 nanometers, much, much smaller hydrogels, right? And you might say, why do I care? And to be honest with you, it's a real good question. The reason that you might care is you perhaps want to influence the differentiation of stem cells from you want to basically differentiate something down an osteoclast or an osteoblast route, or a neuronal route, or an adipocyte type route. And in that instance, the stiffness of the substrate that is supporting those mesenchymal stem cells actually matter, right? So that's sort of where you would use these kinds of materials. And knowing this, we sort of built out a body of work in the Thordarson group about using these hydrogels as an extracellular matrix mimic, right? So all of the cells in your body, all of the cells in your organs, they're all supported by an extracellular matrix, right? That holds the cells in place and just gives them support and growth like any good parent should. And we noticed that, the structure of these gels, we're not the first one to notice this, by the way, but the structure of these peptide hydrogels quite closely mimics the structure of the extracellular matrix. And this is, we started to sort of go down the path of using these gels for cell culture applications, right? Can we use them to grow tumor spheroids? Right? What sorts, can we functionalize these gels with RGD ligands, right? Are they supportive for growing tumor spheroids? Are they supportive for growing different cell types? Can we change the lineage of cells by changing the stiffness of these peptide hydrogels, right? And I started to get more and more involved in the world of biology, right? And as a chemist, I was against that, right? Didn't want any part of it, and I was forced, kicking and screaming, to get into that particular space, right? But it was a good thing. It was definitely a good thing, because I remember standing up when I started working with the dementia guys. I stood up and I gave a similar sort of presentation. I talked through all the structure-function-activity relationship. I talked through the hydrophobicity scale, the hydrophilicity, the changing of the amino acids. I talked through all of the neutron scattering work that we did, and I thought it was, pretty cool. These sort of structure-function relationships. I didn't care. Biologists didn't care, right? I see Mark smirking in the background because I still have this opinion. Biologists care about biology, right? What they cared about was, we've hired you to make new extracellular matrix mimics to mimic dementia in a dish, right? We're going to take the cells, the primary neurons from transgenic mice that express various pathogenic mutations associated with dementia, and instead of using the mice, right, to look at the deficits that are associated on the phenotype scale, right, we want to look at the deficits associated with the cells. If we take those cells and we grow them, right, can we find a way to better screen treatments for dementia, molecules for dementia, other than putting it into an animal? Is there a way that we can bring that screening process forward in vitro, in a 3D cell culture model, right? That was my job. That was my fellowship. They didn't care about structure-activity relationships. And even to this day, we work with a lot of biologists at PYC. I'd say we've got about 65 people in the company, right? 60 of them in Perth. 50 of them are molecular biologists, right? What do you think our molecular biologists care about from a chemistry perspective? Where's the material? When will I be getting the material? How pure is the material? Those are the three questions, right? That's what they care about. So you can give them context and you can give them insight, and that's fine, but unless people are truly interested, and typically they come up to you afterwards and start asking questions, I learned pretty quickly, you got to learn to just simplify things down a little bit, right? You got to play to your audience. You got to know your audience and play to them, right? The most basic question whilst I was in the lab, the dementia lab that I fielded whilst I was there was, can I freeze this buffer? So that's the level, right? That's the chemistry level. So play to those strengths. Okay, so working in the dementia research unit, eventually what I did after about 6 months of killing neurons with amazing efficiency, I actually managed to grow some. So essentially what the guys in the lab use to culture primary neurons, so primary neurons are neurons that are extracted directly from an animal's hippocampus, and then they're plated and we grow them on the substrate. They're grown on a polylysine substrate because they like cationic substrates to adhere onto. So what the neuron does is it sticks initially and sort of sprouts out some little lamellipodia processes, and they grow, and then you eventually form an axon there, and they start to form a network over time, right? So these are stained for beta-3 tubulin, MAP2, and the nuclear stain is DAPI there, so all are sort of neuronal type markers. And what we managed to find was we hit on a couple of peptides that worked actually quite well as substrates for growing primary neurons, right? The advantage that we had with these peptides over PDL is only ever a one-dimensional coating, right? Only ever one-dimensional coating. These guys, we could, in theory, grow up into a gel. They could provide three-dimensional support for these neurons. The neurons can burrow in. You can hold nutrients in the gel that you form. You, in theory, should be able to create a better 3D mimic of the system that you're trying to model, as opposed to neurons on a flat dish. Because cells on a flat dish, I mean, we are not flat dishes, right? Our cells grow in 3 dimensions. Okay, I know a lot of 2-dimensional people though. So in any case, we managed to grow these particular neurons out to, I think this is day 40 that you see here. And so for a lazy chemist like me that's not very good at biology, being able to put neurons on a substrate and put them in the incubator, not worry about changing cell culture media, and just let them grow for 40 days, and you see the processes really start to expand out. That was really nice to see. That's not something that is that achievable with PDL. They start to basically dissolve the coating to an extent, right? So it's the sort of density of the axons and the processes is not as high on PDL. And I'll try and play this video for you. Before I play it, this is probably the coolest experiment that I've done in the academic space. So what we did was we grew neurons on these gels, on these gel films, and we transfected them with a plasmid called GCAMP6. What GCAMP6 does is it fluoresces every time a calcium channel fires. So every time a neuron fires, you may see a pulse of fluorescence. So hopefully you guys can see that. This is early time points, right? So like day 5 or so, the neurons are not that connected and they all fire individually, right? It's not a concerted team effort. We did this again actually when the neurons grew out to day 28 or day 40 or something along those lines. And the result was a bit different. There you go. So they're all connected, right? All those neurons have connected, they form their processes, and they all fire synchronous, right? All in sync, which was pretty neat, I thought. That was about all I achieved in academia, to be honest, because after that, I went to the dark side, as we used to call it, in the academic space. Went to industry, and to be honest with you guys, it was a pretty, 2019 was a fairly rough year. And anyone who's been in academia for a little while can appreciate this, right? Paper rejection after paper rejection after paper rejection. going for a fellowship, getting good comments on the expert assessor for the fellowship, and then sort of the rug being pulled out from under you or whatever it is. So kind of towards the end of 2019, I was starting to question whether the work that we were doing in the lab, whether the group's work was actually good enough. Was it really going to cut the mustard, right? I had an idea that I thought was good. It didn't get funded. You know, what are you going to do? What am I doing with my life here? You know, I had a 1-year-old at home. My wife and I are both from Perth. You're sticking it out over in Sydney. No support, screaming baby, unhappy baby, right? Your world's crashing down. It's all too hard. And I never really thought about it like this before. And then the job at PYC came up. And to be honest with you guys, I never intended on taking the job. Never intended on taking. I just thought I'd dip my toes in a little bit. And I did. Luckily enough, I did the interview. I was over in Perth anyway for my brother's wedding. I popped up to PYC the next day. I did the interview in a crumpled suit. So I obviously went down well enough. And they offered me the role. And I thought, well, I'll take the leap, right? Why not? Why not? We'll move back to Perth. We've got family back in Perth. What's the point in sticking it out for a job, for a position that only 1% of people that ever finish their PhD achieve, right? So it was a bit of a reality check. And I remember in the interview, our CEO, who's still our CEO at the moment, he asked me, what are you worried about moving from academia to industry? What concerns you the most? And I said, the people. Right? Because the people, it wasn't a reflection of the people that I'd met at PYC, but more the people at Macquarie are amazing, right? That lab worked real hard, right? And it had a mentality of everyone's in the trenches together. We're all fighting together to get this paper. We're all going to lean in and help each other out. I didn't know whether I'd still be able to find that level of camaraderie at PYC. And the freedom, the creative freedom to, in an academic space, do whatever you wanted. I was kind of wrong. I was very wrong on both counts. The people at PYC are amazing, and there's a lot of freedom. It's just a different kind of freedom. It's a little bit different in how you apply that sort of creative freedom. And I'll get into that in a little bit. So I guess the lesson here was be proactive. I was lucky. I wasn't someone, my grant hadn't run out, my contract hadn't run out. I still had 3 years to go on the contract. I was in a very, very privileged position at the time, and I absolutely admit that. But I wasn't going anywhere. That was the honest truth. In that department, right, in a medical department, in that group, you know, with the research, it just wasn't going anywhere. So I had to be proactive, make a move. I reached out to people and be informed, right? So be informed is an interesting one, because when I started my first day at PYC, I said, where do you want me to set up in the lab? Where I'm here to make peptides, right? I'm here to make some peptides. And they said to me, we don't actually make any peptides at the moment in the lab. And I thought, I've really stuffed up here, guys. I really should have done a little bit more due diligence during the job process, and I probably should have done a lab tour. But they pointed me to a desk and they said, you sit down over there and you start working on that Gantt chart there, Adam. So that was my introduction to meaningful Gantt charts. I'll get to that a bit later. But I should have been more informed in the process. So anyone who's considering applying for an industry job, I would advise you to be informed, right? Think, what does the company do? Where does the company want to get to? What's the stage of the company? What are their prospects moving forward? And do your own research. Do better than I did, right? I got lucky. PYC is in a great spot now, but you know, I could have just as easily been walking into a lemon and not had the wherewithal to actually know it. So be more informed than I was. What does PYC do? So originally PYC was founded as a company called Phylogika back in the day, right? Founded basically upon a premise of a diverse peptide library. So what the founder here did was he took the genomes of bacteria, archaea, right, and took those genomes and sheared them and displayed them on the coat protein of a phage to create this diverse library. And the selling point there was all of animals, plants, and fungi are located in this part of the phylogenetic tree here. There's all of this diversity that you see here, up here, archaea, bacteria, all of this sequence peptide genome diversity that is not at all captured by mammalian cells, animal cells, fungal cells, right? So the peptide libraries that we have at PYC are very, very diverse. And the selling point a little bit there was, instead of trying to take a random library, why don't we take a focused library? Why don't we take a library that's been fine-tuned by millions of years of evolutionary pressure to be relevant, to be functional, right? Harks back to the initial slide that I showed you on peptides. The utility of peptides is defined by their structure, right? Take peptides whose structure have been honed over millions upon millions of years. Display them and see where you can point them, right? That's kind of what we did back in the early days, and the caveat here is that I wasn't there for the early days, right? So I'm talking about a life that I didn't live. So that's sort of the origin story of PYC there. And there was a little bit of a strategic shift about 5 years ago. We used to do a lot of collaborative work with pharma and point those libraries at various targets and try and solve various problems. But we then, about 5 years or so ago, decided to take control of our own destiny, as it's been said. So we had the peptide library, but we needed to take control a little bit of the cargo that we wanted to deliver here. And that is where Professor Sue Fletcher comes in. So Sue worked a little bit at UWA, now has a position at Murdoch, she's now retired. Really, really brilliant scientist is Sue. Developed a lot of the preclinical data for the now commercial Duchenne's muscular dystrophy antisense oligos that are used to basically save a lot of lives over, since they've been commercial. So absolute sort of ASO oligo biology genius is Sue. We benefit a lot from having her input in the company. We also leveraged a connection with the Lions Eye Institute. So Dr. Fred Chen here, retinal expert at the Lions Eye Institute. And we took our peptide libraries and we took Sue's expertise in oligo design and preclinical experience, and we took Fred's expertise in identification of high-value retinal disease, and we combined them all into a strategy, right? So there's a lot of layers, I guess, to the initial PYC strategy. We target monogenic haploinsufficient diseases, right? And so what that means, monogenic, you have basically, there's a mutation in a single gene that causes a disease, right? What that means is that you have a link between the mutation and the phenotype that manifests. This is different to a disease like dementia or diabetes, where you have multiple factors involved in those particular diseases. This is a one cause, one disease. So if you intervene at the root cause, you have the potential to change the course of the disease, right? So it's a nice strategy from that perspective. Haploinsufficiency means that the expression of a single allele means that you lose function, right? So what you're trying to do is increase the expression, the gene expression, increase protein production up to normal wild type levels, right? We've also selected diseases where overexpression is deleterious to that particular disease. So what that means is that competitive therapies like AAVs are potentially not applicable to rescuing those diseases. So there's a very specific niche that PYC is playing in here, and it has a much higher probability of translation and technical success. I have a placeholder slide which didn't translate to the version that I saved. But anyway, this placeholder slide should feature a timeline which is basically saying we're in a really nice position right now, and well, thankfully for me, I'm pretty sure it's on our website, so you can go and see it on our website, that we have one program in the clinic at the moment, our lead program on retinitis pigmentosa type 11. I'll tell you a little bit about that in the future, or in the next couple of slides. We have another program for autosomal dominant optic atrophy going into the clinic in the second half of this year, and a third program entering the clinic in the first half of 2025. So we're in a really strong, good position. From when I started with the company, we had about 20 folks, and we're a long way away from the clinic. So in 4 years, we're out about 70 at the moment, 70 folks, 3 programs in the clinic within the next 12 months. So it's been a really wild journey. We've learned a lot as a company, I feel. So RP11, retinitis pigmentosa type 11, this is a severe progressive blinding eye disease. It manifests in childhood, and typically the way that people start to notice that they have this disease is they lose their night vision. So at nighttime, they basically can't see, they can't function. And what this means for patients that have this particular disease is they can't do anything in the evenings. You can't go out and drive in the evenings. You have no vision when it's dark. And eventually what happens is you start to lose your peripheral vision, right? Your sort of photoreceptor and your RPE, retinal pigment epithelial cells, start to die off, and your field of vision becomes narrower and narrower and narrower, as you can see in this sort of illustration here. There's no treatments available at the moment for this particular indication. So this is PYC or VP001, which is therapy, is a first-in-class therapy for this particular indication. It's caused by haploinsufficiency of this particular gene, PRPF31, right? So this is the link to the gene mutation to the phenotype that I was telling you about. And we built out all this non-clinical data. I'll tell you a little bit about this towards the end of the presentation, but there's a lot of studies that need to be done, right? But getting a program to the clinic takes a village. We have to define a safe and tolerable dose in the relevant toxicology species, determine the frequency of dosing in those species and model it to humans, and then we get some conviction that the program's going to be successful by using patient-derived cells. So we take cells from patients that have this particular indication, take fibroblasts, reprogram them to retinal organoids, and apply our treatment and see if we can rescue the phenotype that is manifested in those patient cells. And this is just a real quick graph showing that in the unaffected isogenic control patient cells here, you have normal expression of PRPF31, which is the gene here. Upon treatment of our molecule VP001, you get a dose-dependent increase, whereas if you treat those cells with the scrambled oligo, you basically stay at disease levels there. So we have a dose-dependent increase in expression of PRPF31. That's very nice to see. The big measure really is how it relates to patients. Does it make their lives better, right? So that's currently the subject of our clinical trial, which is located in the US in the single ascending dose study there. The next program going into the clinic, which is second half of this year, is another ocular indication, autosomal dominant optic atrophy. We did not pick it because it has the opposite mechanism of action to, I know it looks like I've just inverted the colors on the last presentation. I promise you that is not what I did, and this is actually how the disease manifests. Patients tend to lose their central vision first, and then that widens out, right? So you're losing the function of your retinal ganglion cells. So here, addressable patient population, again, no treatments available commercially for patients with autosomal dominant optic atrophy. There are a couple of treatments in the clinic as well at this point in time. And again, same, right? Haploinsufficiency of the OPA1 gene in retinal ganglion cells in the eye. That's what we're looking to correct with this particular PYC001 molecule. There's a lot of biology on here, so I'm not going to go into it too much, but essentially what I want to communicate to you guys is upon treatment of patient cell with the molecule PYC001, there's a cascade of events that occur, right? So people that have this disease, basically it's a mitochondrial deficiency. So in your cells, what happens for patients is that they have mitochondria that are disorganized, it's not tightly packed, and because the mitochondria, as we all know, is the powerhouse of the cell, it basically manifests as an energetic, a bioenergetic dysfunction. So here we're recovering the structure, we improve the bioenergetics, we improve their respiratory efficiency, and the cells become more resistant to cell death, right? So that, again, in the clinic second half of this year, and that's been a big team effort there. And we've got some really nice images there. You can find all of these presentations on our website there. The really nice thing with this particular program is that in the non-human primate retina, we were able to establish that a single dose of PYC-001, the drug molecule, increases expression of the OPA1 protein in the retinal ganglion cell layer here versus a wild type that you see there. And this is in a wild type primate. So this is a primate that does not have the disease manifestation, has normal levels of this particular protein. All right, great. So, how am I going for time, Terry? Okay? Okay. Okay, no problems. So, I just want to talk to you guys a little bit about the requirements for publication versus regulatory submission. So, I took one of my half-baked journal articles from when I was an academic, and I trawled through the supplementary information, possibly more thoroughly than when I was an academic, and I looked up, actually, what did I report for these peptides? When I made a peptide, what did I report here? White powder, HRMS, the ID by MS, right? Carbon, proton NMR, IR spectra, and the purity by some generic method, right? Some method that was just, a gradient 5 to 95% TFA is a method that if I showed that to our group lead of QC, he'd probably throw a chair at me, right? Because it's not selective enough, not purity indicating. For our current sorts of materials that we're looking at here, it's a bit of a wider spec, right? What the regulatory body wants to know is you need to characterize the absolute bejesus out of this molecule. You need to know everything there is to know about this molecule. You need to be absolutely sure that you know all the details about this molecule that's going to be put into a patient, right? And this isn't even the final form of the molecule. This is the drug substance. This is the powder that's going to get dissolved up into a solution, and that's then going to get injected. So for the powder, appearance is the same, the idea is kind of the same, but for a peptide, you need to know the sequence. What's the sequence of the peptide? That's MS typically. NMR, IR stay the same. The purity is an interesting one because for a GMP material, you need a validated method. So I need to be able to resolve all of the impurities under my main peak. I need a method that separates out all the impurities. A nice example that I can give you guys here is a long time ago, we used to get peptides from a supplier that were 90-95% pure with a 5 to 95% TFA gradient that I would normally run in an academic lab. We then developed a purity-specific method, so one that teased out the impurities, and that purity dropped to about 60%. It was a massive, massive differential. You can hide a lot of impurities, right, with a squished gradient on a HPLC, right? So it needs to be a purity indicating method. You need to report all of your impurities by retention time that are below your limit of quantification. This peptide has a counter ion. If it's a polar peptide, if it's got an arginine, if it's got a lysine, if it's got a glutamic acid, it's got a counter ion there somewhere. How much counter ion does it have? What's the water content? All of the solvents that you use to make that peptide, DMF, NMP, TFA, all of those things, DTT, TIPS, everything you used in that process, quantify them. Metals that could be leached from your reactor, quantify that. Sterility, endotoxin, and chirality, right? So that's fun times, right? And it's definitely, you learn a lot about the molecule, and you learn a lot about the process when you have to go through all the method validation documents. So it's very thorough, right? I guess is the point here. That's like the tiniest part. That's the tiniest part of the regulatory submission, right? That's just a COA. This fun pyramid is basically what your submission to the FDA looks like, the common technical document, the CTD, they call it, right? And you have all of your different modules. So module 2 that's a summary module. There's a whole module that's just summaries of summaries, right? Which sounds insane, I know. Module 3 is kind of where the chemists live. This is our happy place, guys. Module 3, right? You get to tell people a little bit about the process, the in-process controls. How did you control the specification? How did you control the impurities? What were the batches that you made, right? This is our happy place. For a chemist, these are less happy places, but they're still important. So module 4 is all the tox that you did. So you want to dose a molecule in a patient, right? How do you determine what dose that you go in? How do you determine how often you go into a patient? All of your pharmacokinetic work, all of your PD work, all of the establishing the maximum tolerated dose, everything like that, that's module 4. That goes into module 4, right? All the cell-based work, and this is your clinical synopsis. What are you going to do in the clinic? That's module 5, right? And you tie it all together. Module 3, our happy place, guys. That's what it looks like. I don't know if you still think it's a happy place. I can assure you that it is still a happy place, right? This is just the drug product, but there's a lot of things to go in there. There's a lot of data, right? So you've got to know your molecule inside out, back to front. Okay, last slide. Lessons learned, myths busted. At the end of the day, the biggest thing that I noticed when I made the move is that the people are the people, right? Everyone's driven. Everyone's motivated. You can't succeed in the academic space without being motivated, without being driven, right? You're constantly facing rejection, right? And you're living with that rejection with everybody else in the academic space that's in the trenches with you. It's the same thing in biotech, right? You're trying to get a drug to patients. You're all down in the trenches. You're all working towards the same goal. People are driven. They want to help. They're inherently good. Biotech is not a 9-to-5 job. For some reason, don't ask me why, I thought biotech was 9 to 5. How you going, guys? 9 o'clock, I'll punch in, I'll do my work. 5 o'clock, punch out, I'll take my paycheck that's about 3 times bigger than my academic paycheck, and I'll go home. No, not at all, not at all. I do more hours currently than I ever have done as a postdoc or a senior research fellow or something like that. So it can be a 9-to-5 job, depending on your role, but it can also be not a 9-to-5 job, right? Biotech is not the solution to your fiscal problems. I got bad news for you. If you wanted to make a lot of money, science, it's a no, right? So real estate, maybe venture capital or something like that. But we let's be honest, guys, we got into science because we love science, right? So a permanent role in a startup or a biotech does not guarantee job security. People see permanent positions in a biotech. Everyone could be made redundant next week unless you go work for a CSL or a company like that or a Moderna or something along those lines, right? The job security, that permanent position is not always what you think it is. In academia, the most valuable commodity is dollars. You're always trying to scrimp on dollars. We always used to get stuff. We always used to think about buying things, and then the next question was, can't we just make it ourselves? We can make that ourselves, can't we? I got the paper from 1970 in that German journal. Yeah? The one I put it through Google Translate. We can make it. All right, come on, let's make it. That's at least my experience in academia, right? It's the opposite in biotech. You want to what? You want to make that? Are you insane? It's going to cost like $500. Just get it delivered. Just get it shipped, right? The most valuable commodity is time. I don't have time for someone to tool around for 3 weeks trying to make a compound and fail when I can get it in a week from a vendor, even if it's 3 times more expensive. It doesn't particularly bother me, right? If we're talking $400,000, $500,000, $1 million, yeah, okay, different story. But if it's $500, $6,000, time is the most valuable commodity here, right? Time is what our shareholders care about. Yeah, time to patients. The way that we explain it to the new folks is our time is, our shareholders are paying for our time, right? We're a publicly funded company. Everyone that's invested in this company is paying for us to be here right now. They're paying for us to be efficient, right? And our time is best spent getting therapies to patients. That's the overarching goal that we need to keep in mind. So time is really important. Creativity. I talked to you guys about creativity earlier today. I was worried that I wouldn't be able to be creative like I was in an academic lab in industry, in biotech, but it's different forms. Creativity in my job is thinking outside the box in terms of solutions. It's coming up with running things in parallel or coming up with a pathway that we hadn't thought about before, making connections that nobody had thought to try and give our drugs a better chance of technical success in the clinic, right? It's not necessarily determining what diseases we go after. I asked a few of my colleagues, what have they learned? What have you learned from coming from academia into industry? And the 2 responses that came up a lot pragmatism, right? You can't look at things with rose-colored glasses. Things are going to work out, things are going to work out. Because more than likely, we're all scientists here, things don't work out, right? They don't. Science most of the time does not work. And that's okay, yeah? Design the experiment that allows you to assess whether something is going to work, whether it's not going to work, and if it doesn't work, move on. Move on to the next thing. You have more important things to do with your time. Right? So failing fast, really important. You've got to design the right experiment so that you can fail fast, because I'd rather someone fails on an experiment in a week than in 3 months, right? Because that 2.5 months is very, very valuable. All right, I think we're at the end. So I wanted to thank everyone for their attention, and thanks to everyone that's had to put up with me across my journey between Curtin, UNSW, Macquarie, PYC.
Thank you all for coming, and hopefully see you at the next event. Thanks.